All right, next up we have Andrew Miano, recent graduate now, just defended his thesis from SUNY ESF.
Thank you everyone for coming. Today I'm going to present some work I did as part of my master's project, which I recently defended. Most people here probably know about the round goby, but for those of you that don't know, the round goby is a small benthic fish that's native to the Pondo-Caspian Sea region. And normally in the Great Lakes and where it's invaded, we normally think of them as being around between 40 and 150 millimeters in total length. But recently in the St. Lawrence, we've been finding them in excess of 250 millimeters, which is an extremely large goby. You can see a picture of them on the left there. So a brief history of the round gobies in the St. Lawrence River. They were first introduced around 1990 in the St. Clair River, and then by the year 2000 or so, they've been introduced into all the Great Lakes. And by 2002, there were about 9.9 billion round gobies in western Lake Erie alone. So a lot of biomass. And the first time we actually found them in the St. Lawrence River was in around 2005. In 2006, we picked them up in our long-term surveys at the Thousand Islands Biological Station, and their numbers have been continuing to increase all the way into 2015. So this is long-term data that's coming out of the Thousand Islands Biological Station. The top graph is our trap netting survey, which is actually designed to catch muskie and pike. So we use very large nets, but they actually capture very large gobies. The bottom graph is our 30-foot steining survey. It uses very, very small mesh, and it generally captures a lot smaller gobies. But as you can see, we've seen this really great increase in goby populations over time. And 2015, we actually got the highest numbers of both large and small gobies. So these things are still increasing in our area and are still very problematic. So why actually study the round goby? The round goby does a lot of things in ecosystems. They're a known disease vector. They also displace a lot of native organisms, such as sculpins, log perch, and darters. They're also a known egg predator, particularly on cincharchids, lake trout, and also a sausage sling I touched on last year in my presentation. But they also have this great potential to alter our native food webs. They consume a variety of different prey, mussels, diptera, zooplankton, small fish, and even fish eggs. But it's unclear how these diets actually differ by location or by different size classes, particularly the very large size class that we're seeing in the St. Lawrence River. So our research objectives for this were, does location matter? So are different bays behaving differently in the St. Lawrence River? Are gobies consuming different items? And also, does size or length affect round goby diets? What are these really, really large gobies shown here in the red? What are they doing to the system? What are they feeding on? How much is that going to hurt our ecosystems? So in order to answer this question, we collected 40 round gobies from 3 bays in the St. Lawrence River. We collected 20 large gobies, so these are gobies greater than 130 millimeters, all the way up to 235. And then 20 small round gobies, 48 to 129. And these small gobies are what normally people think of as a regular sized goby. The large gobies are extremely large. And then we analyzed all these gobies for length, weight, diet, dry weight and abundance, which is more of a snapshot of what they're feeding on. And then also stable isotope content, which is a little bit more integrated approach to see what they're actually feeding on. For our stomach contents, we sorted them all to the lowest identifiable taxonomic group, dried and weighed each of the items, and then we analyzed them using an MDS ordination, an anasim, pairwise test, and we also did a feeding diagram developed by Evanson et al. Our stable isotopes, we used the same 120 round gobies, took tissue from them, sent them to Cornell Stable Isotope Laboratory, dried, weighed, and run through a mass spec. And these isotopes give us, the two we focused on here are carbon and nitrogen. So nitrogen will actually tell you trophic level. The higher up your animal is feeding on the trophic level, the higher nitrogen value you'll have. And carbon, you are what you eat. So your carbon signature will show what that animal is actually feeding on. So the combination of isotopes and a diet will give a pretty good understanding of what these animals are actually eating. So this is dry weight data for our stomach contents. So these are NDS ordinations. So the way you want to look at these is just if a fish is eating similar to another fish, each of these points is a fish, they'll be very close to each other. And if there's good separation between groups, that means that your 2 groups or 3 groups are eating differently from one another. So our top graph, you can really see that out of our 3 sites, there doesn't really seem to be any differences between the 3 bays that we looked at. And that was confirmed by our anasim as well. A little bit more interestingly is the differences between size masses. The bottom graph there, you can see that there actually is very good clear separation between our large gobies and our small gobies across all of our sites. And our anasim confirmed that yes, that this is significantly different. But that doesn't actually tell you what large gobies and small gobies are actually eating. Why are they coming out differently? This is average percent weights for large gobies versus small gobies. And a couple things I want to highlight here is that large gobies are really consuming a significantly more dry-scented mussels in their diet, while our smaller gobies are consuming significantly more eggs and also chironomids. So there does seem to be some differences that we can pull out right away just from looking at this data. But this won't actually tell you too much about what they're actually selectively feeding on, whether or not they're specializing on a certain thing or generally feeding on it, which is why we used this diagram developed by Edmondson et al. And what this does is this actually tries to plot out your individual prey items and see whether or not a fish such as our goby is actually feeding specially on something or generally on something. And the way it works is you have a frequency of abundance on your x-axis and your prey-specific abundance on your y-axis. And if, say, you have a goby that all your gobies in your sample are eating chironomids, but chironomids in any one diet don't make up that much of any one diet, then it's going to fall out in a dominant but not important on the lower right-hand corner. But if you have a goby that consumed one very large fish, but no other gobies consumed a very large item like that, a fish would come out as important, but it's not actually a dominant thing that they're feeding on. So we're going to use this going forward. So our smaller round gobies, a couple things we can see. So most items are appearing under rare or generalist. So we can kind of think of as small gobies actually appear to be generalist consumers. The one exception is the chironomids, which most gobies ate chironomids, but in any one sample, they weren't a significant portion of that diet. This is a lot different than our large gobies. Our large gobies, you can see, still have a lot of things that are in the rare or generalist category, but dry-scented mussels really come out in the specialist dominant area. So not only were a lot of large gobies consuming dry-scented mussels, but it was also making up a large portion of those gobies' diets. So they're specializing on those, which is a big difference than our small gobies. The other one exception is, you can see BSB is Brooks stickleback, so we actually found a fish inside of one of our large gobies, but it was only in one fish, and it did make up pretty much the whole diet of that fish. So it came out in the upper left-hand corner, so it's important, but not a really dominant prey item. So now that we've got that out of our diet samples, we want to look at our isotope samples and see if this actually agrees. Are round gobies, larger round gobies, actually specializing on dry-scented mussels? This is nitrogen, so this is, you can think of as trophic level. You can see that there really doesn't seem to be too much variation among our sites, but there is this decreasing trend that's significant with larger round goby size. So normally you think of as a larger fish eating higher on the food chain. It's actually the opposite for round gobies. So they're actually feeding lower on our trophic level, which is probably indicative of the fact that they're consuming dreissenid mussels, which are pretty low. What will tell you a little bit more information about what they're feeding on is our carbon. So this is you are what you eat. You can see that really clear trends here that there doesn't really seem to, again, to be much variation among our sites, but between our size classes, you can see that you have this, for our small gobies, it's very, we have a high degree of variability. While our large gobies, it's very truncated. They're all pretty much eating exactly the same thing. And you can see this significant decreasing trend here with increasing round goby length, which plateaus around net minus 22. And published literature and some of our samples as well suggest that minus 22 is about what dreissenids are. So this is a pretty good indication that they are in fact eating on dreissenid mussels. So with taking all these things together, our diet and stable isotopes have, there's very little variation between our sites. So out of our 3 different bay sites, a goby in one bay is going to consume a very similar thing to a goby in a different bay in the St. Lawrence River. But there does seem to be big differences among our 2 size classes. So our large gobies are the ones that are really specializing on our dreissenid mussels, while our large goby, or our small gobies are actually generalists, but they're the ones that are actually consuming the eggs, at least that we found. So what does this actually mean? Small gobies may actually affect our native ecosystems a lot more than larger round gobies, as they're the ones that are consuming a bunch of different prey items and consuming the eggs. But our large gobies are still going to affect our ecosystems. They're still consuming a lot of different prey items, but they're especially going after these dreissenid mussels. And their populations may actually be related to the number of the dreissenid mussels in a system. You may not actually get these very, very large individuals without dreissenid mussels being present. And with that, I want to thank a whole bunch of people. This project wouldn't have been possible without them, and also our financial support. Thanks. I'll take any questions.
Do you have time for questions?
So, switching diet and size is slightly reminiscent of switch to bacillary. And I'm wondering if you looked at, can you hear me all right?
I can hear you a little bit, yeah.
Can you look at gain differences between the small and large categories to see, is the difference that they got big enough to consume the large dreissenids and then they switched over to that diet, or are they just in some other way different strategies?
Yeah, I do think that is a major driver of the difference between the small and the large gobies. The question was about gait limitation and whether or not, as a goby gets a lot larger, whether or not they're feeding more on dreissenid mussels, because they can. I think it may actually be related to the fact that most of our mussels that we actually have left in the St. Lawrence River are the larger quagga mussels, as opposed to our smaller zebra mussels, which have kind of been outcompeted by the quagga mussels. So I think that, yeah, I think the gait limitation is really key. I think that is one of the big reasons. I think once they reach a size that they can consume the dreissenid mussels, they will pretty much exclusively do that.
Yeah, do you have any thoughts on, maybe from 30,000 feet, for the overall management implications, management for fisheries, management for dreissenids, for large ecosystems or small?
For dealing with the round goby?
Yeah, do you encourage it where you want to get rid of dreissenids? Do you discourage it? Do you encourage it for lake trout?
Well, once they're really in the system, there's not too much you can do about it. If you do have a system that ends up getting gobies but doesn't end up getting dreissenid mussels, you may not necessarily have to worry about too many very, very, very large individuals. But I think the management is mostly just preventing their spread, especially in areas that have large number of dreissenid mussels anyway, because that's a very dominant food source for them. So those areas that already have established populations of zebra mussels and quagga mussels, those already have a really good food source for gobies. So that's where you would probably want to stop the spread of them.
Maybe you want to encourage them.
But, or, yeah. Yeah. I mean, so we did find that they are specializing on them, but the large gobies are consuming a lot of other things. They're not just exclusively going after the dry-sinid mussels, even though they are specializing on them. So large gobies are still a big problem, but they do appear to be really keen into those dry-sinid mussels.
Thanks.
Yeah.
I guess sort of watching all these species introductions from afar, one of the things that really struck me with the introduction of zebra mussels was how much productivity was being pulled out of sort of the pelagic zone of the ecosystem and down into the benthic zone. And now you have a species that looks like it's pulling some of that energy, probably a fairly efficient way. And I was really struck by the population numbers in Lake Erie, especially pulling energy from the benthic zone back up and potentially making it available to fish predators. Are there fish predators that feed on gobies, and do you see that as potentially being a major sort of energetic path?
Yeah, so there's a good and a bad with the round gobies. Some of the things I highlighted here were bad, but they're unlocking the benthification that occurred with the zebra mussels and the quagga mussels. And that is good. A lot of fish are feeding on the walleye, bass, are looking down now at round gobies. So they are feeding on them, which is a positive thing. But there is this kind of trade-off, whether they're eating their eggs, they're eating a lot of things in the food web, but they are unlocking nutrients that happen because of the zebra mussels and quagga mussels. So it is definitely a trade-off.
Seems to me like there's a fascinating project there for an energetic modeler in Lake Erie especially.
Yeah.
How did you choose the 130 millimeter size tunnel?
Yeah, so
We saw the main lake, we see the switch over to around 90, 100.
yeah, so most previous studies have really looked at a lot smaller size class. So usually you think about a diet switch occurring around either 75 or 90. We chose 130 because we were really getting good separation with our different capture methods and our different locations. And we also got really good sample size of above 130 and below it. And our initial preliminary tests showed that there did seem to be some sort of separation there. And when we did the isotopes, you can kind of see that at 130 is really where it appears that everything past that was just focused around -22, and everything above that was kind of generally all over the place. And I did look at it in a couple other size class breakups, but nothing really seemed as pronounced as 130.
Liquid average growth rates, how long is it taking from the small size to large size?
To get to the large. I'm not 100% sure on that question. I have pulled some otoliths and looked at them, but it's not as long as you would think. I think it's probably on the order of 5-ish years to get past 130.
All right, I
Thank
think we'll
you.
cap it there. Thank you very much.